So I just want to remind you all, everything on this page was referring to asexual reproduction. Asexual reproduction is where you do not have genetic variability. You produce offspring that are identical to the parent, but it can happen faster, which would sometimes be what we would say the advantage of this type of reproduction is. We talked about some examples in prokaryotic cells. The ones on the screen now are the examples in a eukaryotic cell. You are made of eukaryotic cells, so are plants. We were on the point about spores. Spores are very similar to seeds. They're not the same, which is why I made my equal sign wavy. But a spore is a small little structure that seems kind of like a seed. A spore contains genetic information in a membrane, and sometimes there's a cell wall around it. But it's a tiny little structure that has genetic information. If I was to try to decide what would be a good representation of an organism that produces spores, a mushroom would be my example. So if you were to look at a mushroom, which I'm going to attempt to draw, it's going to be beautiful. Underneath the cap of the mushroom, there's these little, look like little things that can fall out. Those are the spores of the mushroom. So spores can be used for asexual reproduction in eukaryotic organisms. They're very much like a seed, except for that a seed is formed through fertilization. So a seed would be sexual reproduction. A spore resembles a seed, but is used for asexual reproduction. Now, because we got cut off, we probably didn't give anyone a chance to ask some questions about the other types of asexual reproduction. So before I talk about spores a little bit more, which I will do, does anyone have questions that they didn't get to ask yesterday about budding, vegetative reproduction, fragmentation, or parthenogenesis? Yes. You bet. So parthenogenesis is when an unfertilized egg, so one that is haploid, develops into an adult. So the female would lay eggs. They would not get fertilized, but the structure would develop into an adult. So it would grow, it would get larger. The organisms produced through parthenogenesis are generally infertile, so they're not able to reproduce themselves, which would make sense because they don't have the correct number of chromosomes to make gametes the way a normal organism would. And the example of parthenogenesis that I was talking about was bees. The queen bee lays a bunch of eggs. They develop, they become her worker bees, so she's the genetic parent of all of those organisms. I will mention something about fragmentation as well, because someone asked me this question yesterday. Fragmentation has to be the formation of a new organism. I brought up the idea of a starfish because a lot of people know that starfish can regrow limbs. Regrowing a limb is not fragmentation because I haven't made a new organism. If the limb that was cut off regrows a new starfish, that's fragmentation. So I wanted to make sure that that was clear. So just being able to regrow a structure is not fragmentation. In all of these, we're talking about reproduction. So we have to be getting offspring here. There has to be a new organism that results from it. Does anyone have any other questions they'd like to ask about any of these types of asexual reproduction? So we're going to take this idea of spores and we're going to continue it. There is a picture here which you could find in your textbook in a very similar form. In your textbook, you would be looking for page, let me get it here, 577. And there's a similar picture there if you're writing things down and you don't want to recopy this. Basically, what we'll discuss here is the idea of alternating generations. By alternating, we are talking about sexual and asexual reproduction. So some organisms will purposefully, in their life cycle, sometimes reproduce sexually and sometimes reproduce asexually, depending on the point in their life cycle that they're at. Now, there are two terms that are important here that are new. The first term is gametophyte. If you look, it has the word gamete in it. So the gametophyte is the name we would give to the structure, to the organism, when it is in the process of producing gametes. A gametophyte is haploid. So if you look at the picture, it's got a color coding on it, and it's very similar in the textbook. There's a color coding on it as well. But the gametophyte is haploid. It has half the regular number of chromosomes. The gametophyte makes gametes here via, I could write these words here. It makes gametes. Now, since it is already haploid, we're not going to say that meiosis is the direct process that's making them here. It is using mitosis because it's already haploid. So on this picture over here, here's the gametophyte. Now, this is just a very generic representation. The type of organism that would do this type of life cycle very frequently is a plant. So in your textbook, there are examples of a fern, of a pine tree. Those are the really common ones that get used. I've also seen algae be the example for this. So this gametophyte is the organism. This picture is just representing a generic bunch of cells, but we could say this is the actual fern or the actual pine tree. Gametes are always haploid, so they're on the top part of the picture. But at some point, gametes will join together. So you'll notice that here's the gamete that came from this gametophyte. This one is coming from another organism, possibly, but it's a different gamete. It's a gamete from another place. When two gametes join together, this is called fertilization. So after the gametophyte, which is haploid, makes gametes via mitosis, two gametes join, and we call this fertilization. Immediately after fertilization, we would call the structure a zygote, and that's the point where we would say that the number of chromosomes has changed. So notice how the gametes are up here in the green part. They're haploid. The zygote is definitely 2N. It's on the bottom part. It's in the blue section. It's diploid. The sporophyte is the organism that develops from the zygote via mitosis again. So the zygote is just going to get bigger. It's not going to change its number of chromosomes, but it's going to get bigger, and it's going to form the sporophyte. Now again, this would just be the organism at this stage in its life. So this picture is just a generic group of cells, but this would be your fern plant or your pine tree or your algae. The sporophyte will eventually form spores via meiosis. So right here, where blue switches back to green, we would say that meiosis has occurred. Meiosis is the process that reduces the number of chromosomes. Then the spores will go back to the beginning and become the gametophyte. So The gametophyte produces gametes. The sporophyte produces spores. So they're named for what they produce, not what develops into them. And that's an important point to note because sometimes people will mix that up. Now, does anyone want to ask any questions about any of the vocabulary that's on here? So I'm just going to pause. Now, frequently when we talk about alternating sexual cycles, alternating generations, we're talking about plants. But this is an example of the same type of thing happening in an animal. So this example is showing you for a jellyfish, which I think is the best example of this, how at some points in its life it would be diploid, at some points in its life it would be haploid. So if I take a look over here, I have a diploid organism. Now when we're talking about jellyfish, the polyp would be the young version of the jellyfish. Knowing specific jellyfish terminology is not a required part of the curriculum, so I'm just telling you this so you know what we're talking about. This structure then is diploid. It has 2 N chromosomes. So it can reproduce through mitosis, it can grow, it can develop, and become a medusa, which we would say is sort of the mature form of a jellyfish. So we're using asexual reproduction here. We're just making extra copies of the cells that we already have. A medusa could undergo meiosis. So we have medusa 1 and medusa 2. One of them is making an egg and one of them is making a sperm. Those 2 will join together at fertilization and become a zygote. So just like with a plant, an animal like a jellyfish has a portion of its life where it's using mitosis, so asexual reproduction, and a portion of its life where it's using meiosis, so sexual reproduction. You're always looking for meiosis and fertilization. Anything that happens after meiosis is haploid. Anything that happens after fertilization is diploid. Now, the cool thing about a jellyfish is that sometimes the jellyfish can go back and forth between polyp and medusa. Now, this is beyond the scope of something I would ask you on a test. But I thought that perhaps if someone was interested or had heard of this thing called the immortal jellyfish, that would be a good thing to do an article talk about. The jellyfish basically goes back and forth between young and old and theoretically could live forever. There are also lots of really cool underwater organisms that use one of these methods of reproduction that we've talked about. So if you like marine biology, maybe this would be something that interests you for your talk. The first cell that we're discussing is a fly cell. I told a few people over there, if you take bio in university, you will get to work with these stupid little flies. The reason is their eye color is a genetic trait, and they reproduce fast enough that it's easy for you to study them. But they're alive, so they can fly away. It's super irritating. I had to study them. I did not enjoy it. Anyways, these little fruit flies, Drosophila, we're looking at a germ cell. So this is a cell that would eventually be making gametes. And we're looking at it after telophase 1. So after telophase 1, we have reduced the number of chromosomes. So meiosis 1's job is to take diploid cells and make them haploid. So at the end of telophase 1, we would have a haploid number of chromosomes. That means that if I started out with a diploid number of 8, this cell would have 4. Now the other question is, would they be duplicated? Would I still have identical copies of each of them? And the answer is yes. They would be duplicated. The sister chromatids, which is what a duplicated chromosome is, so I'll just put that here. Sister chromatids do not separate until meiosis II. Now my second question is very similar. It's for the P cell, still a germ cell, but this time we're looking at telophase II. After telophase II, the cell will definitely be haploid. So meiosis I reduces the number of chromosomes. Meiosis II does not. In meiosis II, it looks essentially the same as mitosis, where the number of chromosomes stays the same. That means that if my diploid number was 14, my haploid number would be 7. At this point, do I have duplicated chromosomes? No. Meiosis II, after it occurs, the sister chromatids have been pulled apart. Now the next one is the frog cell. It's a somatic cell, so not one that is going to make gametes. And it's an interphase. If a cell is an interphase, it should always be diploid, so 2N, so it should have 26 chromosomes. Now this is the only spot on the table where I think that there is more than one answer. Are the chromosomes duplicated in interphase? The answer could be yes, or it could be no. If you said no, you would be right if the cell was in G1 of interphase, because chromosomes have not yet been replicated. You would be correct to say yes if the cell was in S or G2, because now the chromosomes are being replicated or have been replicated. So a duplicated chromosome does not count as an extra one in the number. It's just a copy. We are only counting how many different chromosomes do we have. We're never counting the identical copies as part of the number of chromosomes that there are. The last one is tricky. The last one is a pine cell, and it is a gametophyte cell. Now we just saw that it is named for the thing that it produces. Gametes have N chromosomes. So if I'm a gametophyte, I should have a haploid number of chromosomes. It's in prophase of mitosis, so I haven't changed the number of chromosomes that it has. So I would say it's N, it's haploid, which means it would have 12. Now, are they duplicated? Yes, because we're in mitosis, and chromosomes always get an additional copy of themselves that's identical, made before mitosis happens. Now, who has questions about something from this table? Really key idea then, in interphase, cells should be diploid. Anywhere in mitosis, whatever number they started with, that's how many they should have. Most of the time, that's diploid. Here, though, because it was a gametophyte, it was already haploid. So you have to look at what the cell started with. Whatever it started with, that's what it should end mitosis with as well. The only time the chromosome number really changes is if the cell has finished meiosis I. If a cell has finished meiosis I, its chromosome number should be half, and it will be half anywhere after that part of meiosis. So at the end of meiosis I or anywhere in meiosis II, the cell should be haploid if it started out as a diploid cell.